Welcome to this presentation on nuclear magnetic resonance spectroscopy, commonly known as NMR. Today we will explore its principles, instrumentation, and powerful applications in chemistry and beyond. NMR spectroscopy is a technique that utilizes the magnetic properties of certain atomic nuclei. It is widely used in chemistry, biology, pharmaceuticals, and even in medical imaging. NMR helps us to determine the structure and environment of atoms inside the molecules. Now we will discuss about its basic principles. Many nuclei such as hydrogen and carbon-13 possess a property called spin. These spinning nuclei behave like tiny magnets. When a sample is placed in a strong magnetic field, these nuclei align either with or against the field. When we apply radio frequency radiation, these nuclei absorb energy and flip their orientation. This is called resonance. As it can be seen in the diagram that by providing the radio frequency radiation, the nuclei absorbs energy and it flips into this position. That is called the resonance. Now we will discuss about the energy levels and resonance. In the magnetic field, a nucleus has two possible energy states, lower energy states and higher energy states. These energy difference between these states determine the radio frequency. This frequency is unique for each chemical environment, and that is why NMR is such a powerful structural tool. It means that each nuclei resonate at specific frequency. Now we will discuss that how the chemical shift is produced and what chemical shift tells us, that how shielded or de-shielded a nucleus is by surrounding electrons. More electrons density means more shielding and an uplift shield. Less electron density means de-shielding and a downfield shift. These shifts are measured in parts per million and we use the reference standard TMS. As it can be seen that low electron density, this nucleus experiences strong magnetic field because it is not shielded as much. As compared to high electron density, this nucleus is shielded and experiences weak magnetic field, and it will appear a field and low energy. Spin-spin coupling. NMR signals can split into multiplets. This splitting occurs because neighboring nuclei influence each other. This results in pattern like doublet, triplet or quartets. The spacing between peaks is called the coupling constant or J value. Now we will discuss about the Key components of the NMR spectrophotometer: a superconducting magnet that provides a strong magnetic field, a radio frequency transmitter, and receiver to excite and detect signals, a probe that holds the sample, and a computer that processes the data into a readable spectrum. Proton NMR is the most common because hydrogen is abundant and is highly sensitive. However, carbon-13 NMR on the other hand is less sensitive due to low natural abundance, but it provides valuable complementary information about carbon environments in molecules. NMR is essential for determining the structure of organic molecules. It is used to identify functional groups, study proteins and DNA, analyze reaction mechanisms, and even monitor purity. In medicine, the same principles are used in MRI scanners to image the human body. NMR offers several advantages as it is non-destructive, highly informative, and can provide quantitative data. However, it is expensive, requires highly specialized equipment, and some nuclei are naturally less sensitive. Thank you for watching the video.